Thank you for coming to my poster. My name is Shinichiro Ono. This poster is about prediction of metastasis based on gene expression in normal tissue adjacent to tumor. Metastasis remains the main cause of tumor-associated death. Knowledge of tumor initiation has been accumulating, as in the case of oncogenes and their application in multi-gene panel testing. However, how metastasis occurs at the genetic level is still unresolved. In recent years, normal tissue adjacent to tumor, NAT, has been drawing attention. In the proximal surroundings of tumor, there are known to be significant morphological and phenotypic changes from healthy tissue. These changes is known as tumor microenvironment. These changes are evident as far as 1 cm away from the tumor margin. Thus, histologically normal tissue more than 2 cm away, namely NNT, has been treated as a control in tumor studies. However, recent studies have shown that the gene expression pattern of this NNT differs from truly normal tissue. In addition, this unique pattern of NNT can be used to increase the prediction accuracy of survival time and recurrence of a tumor alone. But NNT applicability to metastasis is unknown. So in this study, we aim at validate that metastasis prediction based on transcriptome of NNT is more accurate than that of tumor. In our analysis, we obtained bulk RNA-seq data and clinical data from the cancer genome atlas. We used breast cancer, which has the largest samples of NAT. And in this study, due to the number of sample data, distant and lymph node metastases were defined as metastases together. As a result, NAT were 106 samples and primary tumor were 1036 samples. We created 16 prediction models. We created 8 prediction models for each tissue, NAT, and primary tumor, and the last 8 consist of 2 patterns of input genes by 4 machine learning methods. Finally, we compared the accuracy score of the best model in each tissue. The input gene patterns are all genes and differentially expressed genes, DGs. To check the robustness of the prediction, we split the data into training and test data in a ratio of 7:3. The training data are then input various machine learning models. The machine learning methods use the elastic net, random forest, XGBoost neural network. To create models with high accuracy, threefold cross-validation was performed, and each hyperparameter was adjusted using Bayesian optimization. Finally, test data were input each optimized model, and the accuracy was calculated using area under the curve, AUC. The AUC takes values from 0 to 1, with closer to 1 indicating better accuracy. In the NAT, the number of DGs was much fewer, 18. The specific gene names are listed below, with upregulated genes in NAT from metastatic patient indicated in orange and downregulated in green. In the tumor, the number of DGs was 312. KLK3 and ALB were commonly upregulated in metastasis in tumor and NAT. Prediction accuracy is shown in the table. The higher the accuracy of the prediction, the more it is represented in red. The most accurate for each tissue is indicated by a square. Random forest showed the best accuracy based on NAT and elastic net based on tumor. In this analysis, in summary, the prediction accuracy based on NAT was inferior to that based on tumor. range. Looking at the rows of the table, most prediction models showed higher accuracy when the DGs were input rather than when all genes were input. This may be because the DG removed noise that interfered with learning, as various studies have suggested. However, in tumor elastic net, input of all genes showed higher accuracy. This is thought to be because the selection of genes of elastic net model reduced multiple linearity and picked up signals that DG analysis couldn't. In this analysis, the prediction accuracy was lower than in previous studies. Additionally, XGBoost and neural network did not achieve the expected high accuracy. And in XGBoost, only 9 of the 31 parameters were adjusted. In the neural network, the number of samples was too small for the number of layer settings, 3 to 7. So, improvements in hyperparameters may improve AUC. Finally, since the prediction was possible, NAT may be affected by metastasis or contribute to the metastasis. In future analysis, we analyze the genes that contributed to the predictions. Also, test if accuracy can be improved by combining NAD and tumor. Thank you for listening.